and we'll start the last day of the protein structure with AlphaFold workshop. So let's see what we've got now. Okay, so the plan for today is to go over prediction methods that are based on artificial neural networks. I'll say a few words about what those things are. Then I'll give a couple of examples of simple servers that utilize this technology for predicting secondary structure or memory topology, and then finally we'll go to predictions of prediction of three-dimensional structure of proteins with mostly AlphaFold. This will include going over some of the predictions for the sequences that some of you gave me. So that's the plan. So now, starting with where we got yesterday, the key message from yesterday's workshop is that if you know a sequence of the protein, you should be able to tell what's the structure corresponds to it. The reason is the structure, the native conformation of the protein is determined by the sequence. There's for each conformation we can calculate the energy of this conformation, and then the free energy more exactly, and then in native conformation proteins fold in such a way that this free energy is lowest. That's what led us predict the protein structures without any additional information. You can imagine there might be different scenarios. It might be that there's a specific machinery in a cell that somehow manipulates sequences and puts them together, and then the final structure would be determined by how this machinery works. But this is not the case in nature. Protein folds because they spontaneously fold into the lowest, go into lowest energy state. So that's convenient for us. What is not convenient is that first, calculating this energy is complicated. Some of the terms are very hard to evaluate, so we've got problems with just getting the correct numbers for each conformation. And the second part is there's a very large number of those conformations. If you sample those, if you try every one of them in series, it's not going to work. So you've got to come up with the algorithms that speed up the process. And you want to do those things fast because, as I also shown this slide yesterday, there is a roughly 10-to-1, if not larger, overproduction of sequences versus the structures that are determined every year. We are getting swamped by a sequencing data. It is relatively cheap and fast to sequence another genome and get a collection of thousands of new protein sequences. It's much more labor intensive to determine experimentally structures of those. So that's why we would like to predict sequence structures for all those sequences here that were generated every year. The only tiny fraction of structures were generated. So that's the bad part. The good part is that we are getting tremendous amount of sequencing data. We can use it to when predicting the structures, because in general, if two sequences are similar, the structures of those two proteins are gonna be similar. So we can utilize this information and build models of protein structure based on similarity to other structures. When the similarity is very close, then we can use the approach we looked at yesterday, the homology modeling. But for sequences that diverge farther away, we've got to resort to other methods. This is the Rosetta that was used for quite a while before the AlphaFold came into being, and recently the most popular method and the best performing happens to be AlphaFold. So that's once more the difference in a number of sequences versus a number of structures. We're getting altogether hundreds of millions of sequences and only hundreds of thousands of protein structures. So that's the gap we're trying to fill in. Now, the AlphaFold is pretty advanced application of a general methodology that goes under the headline of artificial neural networks. This is a very generic framework that's used to model to predict the very diverse things. It can be used for image analysis, can be used for protein structure prediction, can be used for speech analysis. ChatGPT is another application. So there are very diverse applications of neural networks, but the core methodology is the same, and the idea is taken from biology, from the way the brain is, the nervous system is organized. And as you probably know, the nervous system is built out of neurons in a first approximation, and those are cells that do have several dendrites which serve as input. They receive information about environment, or from the receptors of various sorts, or they might receive information from another neuron that is connected to a dendrite through a synapse. And then once the neuron receives the inputs, then it generates output on an axon, and then axon can have several terminali which are connected either to next neuron in a series, or some sort of effector cell. It could be a secretory gland, it could be a muscle cell, whatever. So the generic idea is there's a set of inputs that result in some sort of output that's passed over to another module that looks like this, and that's how neural networks look like. They're essentially collections of modules that are often referred to as neurons, and they're connected in a network. You're getting one layer which first layer which receives the input. This is collection of those neurons that are fed with our input data. They perform some sort of operation on this input data, and then return a value that's passed to another layer of neurons. Typically the neural networks are organized in layers, so you can have one layer that receives the input. Then there's one or more layers that are not connected to outside world. They are connected only to the preceding layer, and the final layer that receives information from the previous network and provides the output information, the result of the computation. If you look at individual neuron, it's essentially a function. The fancy name for a function. It does have several inputs, starting from 1 all the way to tens or tens of thousands, depending on the architecture, how large and how complicated, how connected is our network of neurons. Each value, each input value is multiplied by a number, by weight. Okay, the x times 1 is added to x2 times w2 and so forth. Everything is summed up. There might be an additional bias term added to the sum, and then once the overall sum is calculated, then it's either returned directly as an output, or it's passed to a so-called activation function that makes that transforms the value into returned by the sum into the actual output. The original activation function used to be the sigmoid. This is a function with defined like this. This is 1 over 1 plus the input value raised to negative exponent. So this exponential function. When plotted on the xy axis, it looks like this for very small, well, I mean, so very negative values of x. The argument of the exponential function is gonna be very large with the plus sign. It means that this value is gonna be very high. So we're dividing 1 over a very large number, so we're getting something very close to 0. So this is what's happening on the left side. When x is very large, so it's a very large positive number, then we're getting a very large negative numbers argument exponential function. So this is going to be a number which is very close to 0. So then this term disappears and we're getting 1 over 1, which is 1. So this is the value on the far right side, and somewhere around 0, we're getting this step transition from 0 to 1. This is the original function that was used over time. There's an entire collection of different variants of functions that behave more or less like sigmoid. They have a one limiting value, typically 0 or -1 on the left side, and either 1 or and 1 on the right side. There's no specific rule why one function is better than the other than they in practical applications work better. So depending on application, might be favorable to use one function versus the other. So this is not something that can be proven formally that you've got to use function like this, because of whatever math behind it is more dictated by practical side. And that's how this complicated neural network works inside. It's just a collection of very simple repeated unit that repeats over and over again. The neuron, which is essentially a function that add inputs with different weights, converts the sum into, according to some sort of activation function, returns the number. Now, the way different networks solve different problems is to come up with a way that the neurons are connected. So you've got to come up with some sort of a structure of the network, and then we've got to find out what are the values of the weights, and this is the second part. Finding out, optimizing the weights is what the neural training of the neural network is. Essentially, what is done is we take a collection of test cases, test data. For each case, we do have a input values, let's say a sequence of our protein or an image, and then or piece of text, and then we know what we want the network to return in case of our protein structure prediction we would like to have as a return the structure. In case of image, we might want to have a neural network is going to tell you if what's in the image. We can try to make a neural network that's gonna look at individual digits and tell you that we do have a 0, 1, 2, 3, and so forth. Let's say 10 different categories of digits. So in each case, we do have no matter what the problem, we do have a set of input data. This is the data we want to analyze, and then for each individual piece of data, we do have the expected result, and then we present we set the input values of the network according to our input data, and we set the output to expected value, and then we've got to calculate, find out what are the parameters. This is typically done by calculating the so-called gradient of the of the output data. It corresponds, if you think about just two parameters somewhere hidden somewhere inside the network. If we calculate the value of our output function, how does it change for when we vary one of the parameters and another parameter? Then if we calculate the values for of the output for those two parameters, we're gonna get some sort of a surface. We're gonna see how the output changes when we vary the first parameter. So those are going to be lines going from left to right here for each particular value of the second parameter, and vice versa. When we fix the first parameter, let's say 0.4, if we go and start varying the other one, we're gonna get another series of values. Overall, we're gonna end up with some sort of a surface. The way to find where the value is gonna be the smallest is we just start from whatever initial values we come up with for the starting parameters. We look for direction where the slope is steepest going downhill. We go a little bit along in this direction. Once we stop, we recalculate the gradient, recalculate. We look around once more to find out where the slope is steepest pointing down, and we walk another step, and ultimately we might end up in the lowest at the lowest value. So then we stop, and we've got the best version of the theta parameter, and theta 1 and theta 0 parameters. So and this is done repeatedly for many data points and all the parameters. So ultimately we should be able to get the weights for all the connections within the network. The hope is that once we do it for the training set, if we then present a network with a data that it didn't use for training, the result will correspond to whatever we expect for the unknown for the case where the data was not used for training. And that's a very generic description of what's happening with the neural nets in particular cases. Oh, one more thing. So as you see, the training is essentially a repeated application of a unit step which corresponds to calculate the gradient, move a little bit, calculate the gradient, move a little bit along the gradient line, and so forth. So each time you calculate gradient, you've got to calculate derivatives of of the function. You've got to calculate the slope, so that's corresponding to calculating derivatives. So one of the driving forces behind coming up with all those various activation functions is that they are either faster to calculate the gradient, or they the overall procedure is more is behaving better when you're running the gradient descent method. It's mostly because some of the numerical procedures are either less accurate or more prone to tiny variation in parameters. So some of those activation functions are behaving and giving returning results faster than the others. So this is to a large extent experimental. You can try people when designing neural network systems. They can try different activation functions, different architectures, and different ways of training data to get the optimal performance. But otherwise, it's very generic. It doesn't matter what type of input data you've got and what's the expected output. So the framework is very generic. It can solve a very wide range of problems. Inside every problem is converted into numbers. So the initial step of designing a neural network is to decide how the input data is going to be converted into a format that the network is going to be able to analyze, and then from then on, it's very generic. Internally, it's the same no matter what the actual problem is. Now, the latest resurgence of the neural net methods, it's possible because it happens that initially, essentially by coincidence, it turned out that the hardware used in graphics cards is optimized for exactly the type of mathematical operations that are performed when training the networks. The operations are essentially multiplying and adding matrices. The architecture of the network is very regular and can be represented as a series of matrices, and then to either calculate the output of a network or calculate the gradient or calculate or move along the gradient. All those operations can be generalized to just two operations: multiplying two matrices and adding those. This is what graphics cards are doing when generating images. So at a certain moment, people in neural network community realize that you can employ the hardware that's normally used for drawing on the screen to actually perform mathematical calculations, and that's the major driving force behind what you see nowadays happening with all the artificial intelligence methods that are mostly based on neural networks, and this is driven by progress on the hardware side. And then the other part of it is the availability of the data. And in case of sequence structure predictions, we don't have problems with the data. We're getting hundreds of millions of protein sequences. So the data is there, and the neural nets allows us to analyze the data and get some sensible predictions. Now, the bad part of the neural nets is that they do provide you a result, but they don't explain how did you get the result. So in a sense, they are sort of a black box. You put your sequence in, you're getting the result out in terms of structure, but the network is not gonna tell you why the structure looks like. It's not like in a case of a physics-based potential function that tells you how to calculate the energy of a given conformation. It's here you do know that there's a Coulomb law which tells you what's the strength of interaction between two charges. In case of neural networks, there's no loss that you're discovering. You've got a black box which takes the input on one side and returns the result, and that's about it. So that's some sort of a limitation, and it means that besides the AlphaFold, you still do have to figure out why and what are details of protein folding. That's what are the actual rules and how this thing works in a sensible way, in a rational way. Neural network predictions alpha is gonna tell you what's the final structure, how it should look like, but that's about it. And then the second part of the neural network problems is or issues is that training is expensive. It requires essentially cutting edge hardware. You're not gonna be able to do much on a laptop with just a generic graphics card. It's you typically need a pretty pricey and power consuming GPUs that are typically in a consumer market referred to as a gamers GPUs. Those are cards that run somewhere in the high hundreds, if not a couple of thousands of dollars. So that's one issue. And the second issue is that training is gonna take time. So it's not like you're gonna be able to train your network in a few minutes. Sometimes it is gonna take days, if not months. In case of AlphaFold, the typical training run takes about a week or so. So that's the time frame of when you're actually designing the networks. For generating predictions is faster, but it's still significant amount of time. So and you do need a pretty advanced hardware to do this thing. So that's one of the drawbacks. And the second drawback is that you need tons of data. You do need copious amount of data to actually optimize your network so they work in a sensible way, and then depending on a problem, it might be possible or not. So it's problem specific. If you read news announcements about various chat GPT networks and whatever is there, some of those models, if you check what was the cost of training, the cost goes into tens of millions of dollars. So that's something that's outside of the realm of academic research. Some of those things are feasible only because the research is backed up by large companies. So that's the bad part of the neural network research. It's resource intensive and requires tons of data to analyze. So that's good and bad about neural nets. Now, in our case, what could be the input data? Well, the simplest case is we can take a sequence. We can take just a sequence of DNA or sequence of RNA or sequence of protein. This is the simplest possible input. It's got to be somehow converted into numbers. We neural networks don't know what the amino acid symbols are, so you've got to somehow convert it into numbers. But otherwise it's gonna be a series of numbers that specify that we've got that corresponds to the sequence of our protein or our gene. Instead of just a single sequence, we can generate a alignment of sequences. Those will be corresponding to proteins in our case that are of similar structure, so there should be some information hidden there in the alignment that provides you with additional information how the protein structure is going to look like. And we can either use entire full version of sequence alignment, so we can feed the network not with a single sequence, but with entire array of sequences with several sequences. Each one might have a different residue at the same position. So we can enter entire block of amino acid identities there, or we can collapse this thing into so-called profiles or position-specific scoring tables, which is essentially for each position a column of numbers that convey information how frequently a given residue shows up in alignment. So for this column, the K lysine would be assigned a number corresponding to higher frequency of showing up in at this column. P proline would be less, would get the number indicating the it shows up less frequently, and then E would be even less frequent. Other residues would be noted as not showing up at all. And the same procedure is repeated for second position. I mean, first position, second position, third, and so forth. So you can collapse this type of full sequence alignment into just a column of numbers corresponding to whatever is encountered in the first position, second position, and so forth. And depending on application, depending on particular problem, one might start either from the sequence alone or provide information on input as a full version of sequence alignment or just the profile. In case of AlphaFold, it's actually all of the above. AlphaFold as input takes both the sequence that you want to predict information on, predict the structure for. It generates it uses the full version of the multiple sequence alignment, and it also converts the multiple sequence alignment into a profile and uses all those three things at the same time. So this is extreme case. It does help. That's one of the reasons alpha is way better than anything else that's there. But the cost is that you need a lot of memory to actually store all of the information. So there are trade-offs between how much you provide information, how much information you provide on the input, and how good the performance is, and what are the hardware requirements. So that's in case of our inputs. Once more, the goal here, the initial goal, is to convert the data that we've got in our case, that the protein sequence and sequence alignments, into something that the network can take, which is essentially a series of numbers. So there's got to be some sort of way of conversion that's got to be devised. And that's the very basics of what neural networks do. We'll make a break for like 5 minutes or so, and then I'll stop and pause the record. Okay, let's. Oh, I didn't record. I think I didn't record. Darn it. Let's share screen and let's continue on. Okay, so Here's a quick run through the first two servers that use the internally neural networks. The first one is Cypred. That's how the website looks like. The way to use it is relatively straightforward. The website is providing access to many different servers. We're interested only in a Cypred, so we've got to check. We want to run Cypred prediction, and then somewhere down the page there's a place to enter our sequence. I'm gonna use one of the sequences that I've got from one of you for running alpha on it. So I'm gonna just take the sequence and paste it into our server website. That's our sequence, and then we can give it let's say my try, or we can actually give job name the name of the sequence so that we know who's who. Okay, so now we put the name here, and the email is optional. This is gonna take just a moment, so we can submit our print. Oh no, they force me to give okay. I can read anonymously well. Okay, let's submit. Do we need anything else? Nothing. Okay, so here's the program running. It shouldn't take too much time. We'll see what happens, but while it's running, we can also run the same sequence through the other server. This is the transmembrane helix prediction. The sequence gonna be the same, so I'm gonna just use the one we've got here. Once more, I can copy it, highlight it, and paste and paste and paste here. Okay, I think I don't need a name, but if we want a FASTA format, we can always take the identifier, the same one as before, and paste it here. Okay, or we can upload the file if needed. With one sequence, probably the header is not needed. If there's more than one sequence, then you obviously got to separate them with the headers, and then we can run this thing. We'll see how the servers are doing. We can see the progress in a log window. With the Cypred, it's still happening. So we'll see how long is it gonna take. It tells us it's a few seconds, but apparently takes more. So I could be that the site is not working. We'll see. Cypred is notorious for not being up all the time. So we'll see what happens. For the deep transmembrane prediction, we already do have the results, and this is what you've seen before. This is a predicted topology in a membrane with transmembrane helices, loops outside or inside, and this is either drawing with just the most likely topology or a plot of probabilities, and you can get more information here in different formats. You can get just a series of characters corresponding to localization aligned with our sequence, or you can get the values somewhere here, provided I didn't mess up anything. Well, let's try it here. Let's see. Okay, you've got to download it actually as a file, and then if you check what's here, that's essentially we can open it in a open it in a ah can open it in a Excel, and we're getting the all the numbers that we need. There's a column with the position and the residue type. This is probably not the best way Excel treated the file, so there's probably a better way of splitting it into columns. But then we're getting numbers for probabilities corresponding to localization of a given residue in a periplasm membrane, inside, outside, and so forth. This is in case of bacterial proteins. There's additional state where you predict if located in periplasmic space in bacterial cells. So you're getting columns of numbers, and then you can generate plots on your own or use them uh, for for other purposes, for further computation. So that's what's coming out of the transmembrane prediction server. It's the usage is pretty easy. We can try another sequence. We can clear everything that we've got here, and we can use another sequence. Let's pick up another sequence that I was given. So here's another one. When we use this sequence here and plug it into prediction, we can run it. Shouldn't take much time. Let's see what Psypred is doing. Psypred, it looks like it's stuck. It should be much faster. It could be that then the server is down. So there's nothing here. It's typical case with the Psypred. So we've got to wait as long as the prediction here. We've got the prediction already present. Now we don't have any transmembrane helices. We've got only prediction that localizes the protein outside of the cell in extracellular space. But now we do have a prediction of a signal sequence. So in this particular case, we can infer that whatever the structure of the protein is, it's not a membrane protein, but it's a protein that's exported outside, secreted courtesy of a signal peptide. So once more, the plot here is just most likely topology. Well, everything is sitting outside, so there's sort of topology, everything is outside, but there's also information. There's a signal sequence, or you can have actual numbers with, as you've seen before, with the probabilities of given residue being either inside, outside, and part of a signal peptide. So those two servers are relatively simple to use. Both are internally implemented as a neural networks, and they provide you information about on a per residue basis. Each residue is classified as localized in a given cellular compartment, or within the membrane, or taking a particular secondary structure. This sadly didn't complete. So I think Cypred is having some issues with actually being able to use, but at least there's a try. Okay, so that's how you run the simple things. Now, as far as AlphaFold goes, there are different ways of either using the already made predictions or generating predictions. If you want to use the predictions that are already made, we can always go convenient starting point is UniProt, we can go to UniProt and find the sequence we're interested in. Here's, for example, this guy. So I'm gonna use those are identifiers from UniProt, so I can use them to find my protein in UniProt. That's the sequence. If I go here, we go to the corresponding record in UniProt, and then we can go to structure. If they are experimental structures, you're gonna see them listed here, and then you might lose interest in looking at the prediction, but you're still interested in, we can go to AlphaFold down here, and then we can select the prediction here, and this is what prediction from AlphaFold looks like. This is pre-calculated prediction that the UniProt website is providing access to. The limit information here is limited. You're only getting the 3D structure with the quality score plotted as a color along the chain with the legend here, as I mentioned before, the high quality locations or locations predicted with high quality are marked in dark blue. Cyan is intermediate quality, and then low quality is yellow or orange. If you want to get a little bit more information, you can always go to the AlphaFold server, okay? And if you click here, you're gonna be transferred to whatever is accessible on the AlphaFold server for this particular prediction. And here we're gonna see more. Here we do get the predicted aligned error, which this is predicted aligned error is not here. Yeah, well, yeah. So we're getting the formal name is predicted aligned error. The practically what it is, it's a way of estimating how accurate the distance estimation is. Because of the way it's calculated, this matrix is not symmetric. So if you look at, let's say, distance between position 50 and 25 here, so intersection here, it's bright green. But if we go now to corresponding distance 25 along the vertical axis and 50 along the horizontal, the color here is different than after flipping the axis. So this score is not symmetric, but it roughly corresponds to how good the prediction of distance and relative orientation of two residues are. And the darker the color, the better. And the rectangular blocks mark things that are predicted with a high confidence. So that's what we've got here. That's the prediction with a little bit more of information, but we're still not getting everything that's relevant. To get full supplement of quality information, you do have to run predictions on your own, and then you're gonna get entire set of additional information. To run predictions on your own, you can use two sites. Both are running on Colab. This is essentially a cluster cluster made available by Google, where the AlphaFold programs can be run. Okay, and there are two versions of those. I gave the links in a slide that I'll post later on a Google Drive, so you'll have the links for both of those. This site, this set of scripts is made available by the DeepMind, by the AlphaFold people. And essentially, the way to use it is to fill in the to fill in a sequence, fill in the field here with your sequence. If you got only one protein chain, then you enter it once. If you're looking for protein-protein interactions or for homoligomers of the same chain, you might enter more sequences here. Each sequence goes into its own box, and then you've got to run individual somewhere here. You've got to run the blocks. This is typical Jupyter notebook with separate blocks doing separate cells doing different things. But to run this thing first, you've got to connect to a node on a cluster to the actual server that's gonna do computations, and that's done here. So you've got to connect. Yes. Oh, well, I can add. It is in the slides, but I'm gonna. Okay, so just to make it simple, I'll just take this link. Okay, and I'll paste it in a chat. Copy. And okay, so this is a link to the AlphaFold version of the predictions. Okay, and then if I'm edit, there's another variant here. This is a alternative in a way more useful that I'm gonna put it here, and this is the other link. Okay, so those are the two links in the chat. Okay, so to run those things first, you've got to connect to a to actual node on a cluster provided by Google. So you've got to connect to something. Okay, once you connect, you're gonna get information. You're connected to a node with certain amount of memory and then a certain amount of disk space. Those nodes should have GPU available, so it's worth to, before connecting, changing runtime type to one of the GPUs. The preferred one is 800, but it's available only if you actually pay into the system. This is free to use if for without any payments if you're using nodes of that are less powerful for things that are more powerful. You've got to pay in for this particular case. I just spent $10 on I paid into account. I paid for so-called 100 compute credits or whatever they call it, and this provides you access to about to several hours of computational time using one of those nodes. I'll try to get 100, but I think I'm not gonna get it. That's the sad reality. I can save it here. I can try to connect. I'm gonna get information. I can't get a 100. I can only get a less powerful node called L4, type of node called L4. So that's fine. Okay, I do have GPU. Okay, now I've got information that my node is connected to a GPU. Okay, I'm getting also more RAM and more disk space than I had before in a generic case. And then once I'm connected, I still do have a no, my sequence disappeared because I reconnected, so I can try to get the I'll get this sequence here and paste it, paste it in. Okay, and for one sequence, that's all that's needed. So now I think we can run all, and then there's a warning because yes, was not authored by Google, it was authored by DeepMind. So there's a warning here. That's fine. We can run it anyway, and this thing is gonna keep running, keep going through one step at a time. This process is slow. This process is slow, and to make it worse, every now and then the page is gonna pop up reCAPTCHA to make sure that you're actually watching it. So it's pretty tedious. It's good if you try one or two sequences that are not too long and prediction doesn't take much time. But for all the practical purposes, you've got more things to run. It's gonna become very rapidly pretty tedious. Still, this is the original published code. This is exactly what's published in the paper. So this is a reference implementation of the AlphaFold. It's gonna go through steps. Initially, it's got to actually download all the pieces of software into your node, and then run sequence alignment somewhere down here, and then run actual prediction. It takes the most of the time in my hands is spent on generating multiple sequence alignment. Here it can take a couple of hours to just generate MSA. The whole process might take a few hours. So that's it's there, but it's sort of clunky. The other version of the public server is this AlphaFold 2. This is alternative implementation of what AlphaFold does. The changes in the way the sequence alignment is generated. Here you're running sequence alignment on a Google node. It's relatively accurate method of generating multiple sequence alignment. It takes time, it can take a few hours to generate just a single sequence alignment. In the case of the other implementation here, the code that runs sequence alignment is not run on Google. People who provided this server, this is in particular Steinegger lab, they do have their own server that's optimized for generating multiple sequence alignments. They've got the server is pretty powerful. It's I think it's got about 1 terabyte of RAM just to store all the sequences in RAM instead of running them, loading them into RAM every time you run sequence alignment. The issue here is that to generate sequence alignment, you've got to search through a database of hundreds of millions of sequences, and they take space. So just reading them every time you run a sequence alignment takes time. So the server they've set up reads those sequences once, and then each MSA generation is way faster because of that. But this is at the cost of having a server with a terabyte of memory just to compare it with what you've got in your laptop. You've got probably 8 to 16 gigabytes. Here you've got probably orders of magnitude more memory in a server. So that's why it's it's faster, and in practice, the generating sequence alignment using this code takes several seconds, not couple of hours. So this implementation is faster because of that. The step that follows the neural network that predicts the structure is the same as here. So this is equivalent in terms of actual neural network underlying the prediction. The difference is in the quality of the and how rapid the alignment is generated. The this method is a little less accurate than the one used by AlphaFold. If we check what's happening with AlphaFold, you see here we're going after MSA. The estimate is gonna take about 15 or so minutes to get the alignment, and this is pretty short sequence. So the longer the sequence, the longer it's gonna take to generate the alignment. So that's what's happening. I'm gonna leave it running and might complete, might not, but that's how it looks like if you're using the public resources. It holds for both of those, that every now and then you're gonna see reCAPTCHA showing up, and you've got to react. If not, it's gonna deem that you're not running this page interactive. It's gonna kick you out. So that's the limitation of the public servers. Once more, I mean, they are fully functional. Otherwise, you can load your sequences and then try to run the predictions, and it's perfectly fine, except that it might be quite frustrating. You might have problems getting access to fast enough GPU nodes, and you might have problems with the page going resetting itself because you're not there. So that's what's there, and this is probably the simplest way to run predictions on your own. The predictions will not be, you won't be able to run the code on your laptop, because it's requiring, because it requires quite strong GPU. Unless you've got a laptop geared towards gaming, then the graphics card might be good enough. Both of those scripts can be installed locally, but the hardware requirements are pretty tough. So those are limitations. So in our case, in case of AlphaFold predictions that are run here in Boyer Hall, we do have the code installed locally, and then what I did with few sequences that you guys asked about. So let's see what came out, and this is gonna be example of whatever you're getting when you've got access to the server, and you run predictions on your own. So what happens, submission is easy. Submission of the code is easy. You essentially create a file like here. This is a file that was used for running one set of predictions. Is essentially a FASTA formatted file with a header for each sequence, or a group sequences denoting what it is. This is completely arbitrary. This can be name you come up with. In this particular case, I used the identifier of the sequence from the UniProt, but it's this can be anything you want. So this is the case of a single sequence that we're trying to predict the structure of. Here's another single sequence. If you want to predict in the structure of a complex between formed by two or more sequences, then you just separate the sequences by colons. So here's a colon separating the first sequence from the second sequence. So here's this block here corresponds to predicting interaction between two protein chains. And here's one more sequence that's is to be predicted on one on its own. Here's another one, and here's a combination, so forth. So, and this is how the input data looks like. You cannot provide more information here. You've got some sort of experimental information indicating, providing some information about the structure. This is outside of the scope of AlphaFold. All you can do is to predict, just provide the sequences, or if you want to generate multiple sequence alignment on your own, you can feed the code with the program with the multiple sequence alignment that you generate outside of the on your own. So this is another option, but that's what Alpha takes, either a sequence or a list of sequences for predicting structure of complexes, or a multiple sequence alignment if you generate it on your own. And then once you've got a file like this, you essentially uh, run a script by specifying the name of the script, name of the file with the sequences, and you specify directory where the results should go. This is going to run for various amount of time, depending on how many sequences you've got there. But at the very end, you're going to see results in a directory where you tell it to drop the results to, and we can look what's there. So let's check here. The directory here was, I'll try to run this thing first. This is the file I was looking at. So let's look here. And so that's not here. Not this is what I wanted. No, that's not. No. That's wrong. Zoom is in the way that what I want. I want this one, and I want directory here. Okay, and we've got a lot of files that were generated. If we sort by name, we're gonna see where I am in the wrong place. No, in the wrong place. Here. String. Okay, I'm finally right place. Okay, so here's the directory that the script generated. If you look at the file names, there's gonna be a block of files starting with the same name, with the name starting the same way, each corresponding to one particular prediction. Okay, let's look first for a single sequence. Let's say here's one of the predictions for a single chain. This is the name of the protein uniprot identifier, and then various files. What we do get here is, okay, we're getting first access to how the multiple sequence alignment looks like. So this is this file named ending with coverage. If we look here, we do have essentially a plot of some schematic of the sequence alignment. The positions along your sequence are along horizontal axis. This is sequence that's about 120, 130 residues long. So that's those are positions along the protein sequence. And then along the vertical axis, each line corresponds to a single sequence aligned to our sequence. And the closer, the more similar the sequences, the higher up is plotted with and the color scale corresponds to sequence similarity, so our sequence identity. So things at the top are very close in sequence, things going down vary, and we're getting, as you can see here, a region that has close to 1200 sequences aligned, and then we've got another block with less sequences. That's right. There's a lot of sequences here that are missing, and then there's another block that's has even less sequences available. So as you see, it's not always that we do have a lot of sequences aligned to entire length of the protein. It's not surprising. It might be that this block corresponds to one particular domain, one particular folding unit that's present over and over within many protein sequences, but then the rest is not there. So if the rest of the sequences don't have those this part of sequence, you're gonna get empty space, and you can see things stacked, marked as line, or just overall count of the sequences provided. So this is one piece of information that you're gonna get. Then you're gonna get actual structures. Okay, those are the PDB files, and by default you're gonna get five different predictions ranked by quality of the prediction. We can look at those with the Chimera. So let's get Chimera. Let's close the session here, and then we can go to file open, and we're looking for the MCR here, and I did look for I did look at this first thing here. So we want to get the PDB file. I make it longer, so those are PDB files. That's what we're getting out, and here's the first prediction. Here we can open it, and that's the prediction. It looks like a noodle. Okay, there's a little bit of secondary structure here. There's a little bit of secondary structure here. The rest is not folded in any sensible way. Now we can color this prediction by the score that you see normally on UniProt site or DeepMind site, by specifying that we want to color, I think, B factor balance. That's the magic command. Essentially, what we're saying is we're telling Chimera is we need to color the residues using B factors. You might remember from the first day that B factors is one of the columns in a PDB file that's normally used to reflect how ordered the residue is. The atom is in a crystal, but here it's used for providing information how good the quality of prediction is. So we're coloring the chain by B factor, and we're using a palette that was pre-made, which corresponds to the coloring scheme of the AlphaFold. So you run this thing. Here's the coloring scheme that we've got here, and what we see here, there's a small part of the protein that seems to be folded in a somewhat reasonable way, and then there's another end somewhere here that looks like an alpha helix. Everything else is of a dubious quality. Now, very often the quality of the prediction score correlates with the quality of the alignment. if we check here, this is the N terminal, and that's right, this is the first methionine here. So this is C terminus end. If you check, if you compare what we see here in terms of color with the with what we see here, I'll try to I'll try to show it here. So as you see, the last 30 or so residues here correspond to correspond to a large number of sequences that are aligned to our target sequence, and then the number of aligned sequences drops quite rapidly to about 1200, maybe 500 or so. So this is quite typical. If the number of sequences aligned to the target goes below 500 or so, the quality of the prediction, the scores are starting to go down. This is a reflection of the of what alpha is doing. Alpha is looking at correlation in mutations at different positions, and you can get this correlation only if there's sufficient number of sequences that are aligned. If the number drops below 500, 400, then quality hurts because there's not enough data that alpha can work on. So that's why at least to some extent this region is not predicted with high accuracy. The tip here is sort of interesting. The quality seems to go up, but if we feed this sequence into the server we used before for predicting the membrane topology, this is actually the region that's predicted to be a signal peptide. So this is what we've seen. This is what we've seen. No, not here. This is what we've seen. This is what we've seen here. Actually, this is the same sequence as used for AlphaFold, and we see that deep TMHMM is predicting the first one you saw residues to be a signal peptide, and that's what we see here. The rest is probably clipped off after secretion. So those things are actually pieces of the sequence that are not present in the final version of the protein. So no wonder AlphaFold can't predict them. So here's one prediction. We can also read the other 4 versions. We can open additional versions of this predictions for this sequence. So we tried so far one. We can pick up another 4, open them together. As you see now, we're getting things pretty misaligned in a space. So as yesterday, we can go to tools and go to structure analysis, matchmaker, use first one as a reference. The other 4 will be aligned to the first one. If we apply, we're getting something like this. So now we're better, but apparently those mispredicted, those noodles, every time predictions run, they look in a different way. They are shaped in slightly different way. So things are misaligned. So we can do now to fix it. We can remove the first I know 60, 70 residues. So let's remove the leading the N-terminal part of the residues. We can do this thing by by running delete. We don't care about anything but the residue number, so we're gonna go, let's delete something from 1 to, let's say, 50 and see what happens. Okay, so we removed initial 50 things. We can go, let's say, 70 maybe, let's do 65 maybe. Okay, we can more, let's do 6. Okay, so now things look roughly nearby. So now we can improve the alignment by going to structure analysis matchmaker once more, the same, and now apply. Okay, close. And now what we can see is that all five predictions are reporting the same structure. The fold here is very similar between the different runs. We can try to trim the whatever is left here even more. If I hover over one of the positions here, it's gonna tell me, it shouldn't tell me what it is. Even doesn't tell me. What I can do is I can just color, let's color, position color, let's color AD red, and we'll see where AD is. AD is here, so we can move even farther. Let's make 95 maybe. Where's 95? Oh, because I've got a typo, so it doesn't work. Here's 95, so this is too far. So let's remove everything to position 90. So that's here. Okay, so here's our final version of a predicted structure for our sequence. We can improve the alignment by going to structure analysis matchmaker and apply. So now we're getting even better. We close the window. This is so far just the backbone. We can show the side chains, and we'll see that they are in a pretty similar conformations. There's a little bit of difference between predictions, but those are residues on the surface. This is an arginine that's exposed to the surface of the molecule, so it might wiggle around. Here's another arginine also flipping around at the end. This is prediction that was not run through the molecular dynamics or any sort of refinement. So those models can be run and improved by using them as a starting point for optimizing the structure using either molecular dynamics or Rosetta programs, and prediction can be better. What other interesting you might notice here is that now let's color this thing by let's color it. We can now color everything gray. Let's color everything the same way. Okay, and then let's look at heteroatoms. Okay, what you're gonna notice is that we've got the cysteine on one beta strand next to a cysteine on another one. This is most likely a disulfide bond. Those are two cysteines that actually make a covalent bond here, making stabilizing this particular structure. Same thing here. You've got another pair disulfide of cysteines forming another disulfide, and here's another one, and here's another one. So here's another one. So this is indication that prediction is quite good. It actually paired out the disulfide, paired out cysteines to reflect the disulfide bonds, which is sort of reassuring. And it also shows you how very small sequences are stabilized to fold into discrete domains. It's very often made by disulfide. So here's example of a pretty good prediction of a small domain. Now let's go to another prediction. Let's close this session and let's go after another one. Let's go and open another set of predictions. Now I'm gonna take whatever I had before. This is one of those ASIP things and the other protein. So this is now gonna be a pair of sequences. Yeah, pair of sequences. So let's look at, let's look first at the coverage. Okay, so at the sequence, but this is gonna be done just this way. Not the, okay, no, cancel, no, I've got to do it from here. So let's get it quickly here. This is gonna be this one here. Okay, when we open here, now we've got two sequences. They are plotted here. This is the one that we looked before, this 130 or so residue long sequence of the shorter protein. This is, we've seen this plot before. And then here we've got another alignment now for the other protein. Here there's not so many predictions, less alignments, maybe 1500, less than 1500, but they cover most of the line sequences cover the full length. There's a short region here that's present in additional sequences, but most of line sequences are covering the entire sequence. So this tells us that there's a hope to get a decent prediction for a full length of the protein, or most of it. And if we actually look at one of those predictions, so if we go here once more, I've got open here and go here, then if I open, so now here's our prediction. Once more we can take and color it according to the prediction quality. This is the B factor palette alpha. Now we do have a similar situation. Most of the protein is folded, but there are pieces that are unfolded. This thing is reminiscent of what we've seen before. This is most likely N-terminus part. If I hover over somewhere here, yeah, this is residue 6. So this is N-terminal part, and if the sequence is fed into transmembrane region prediction, you're gonna see that this is likely a signal peptide, and then the other helices would be identified as transmembrane. This is a clearly a transmembrane protein or membrane protein with helices going across the membrane. To clean this thing up, we can remove the extra tails, but before that, I'll read the remaining structures to see how reproducible the prediction is. So we can open the remaining force drive. Read this one, so now I'm gonna read the remainder. Okay, they are orientation is messed up, so we can go to Tools, Structure Analysis, Matchmaker first. This is default and apply. And once more, we've got the same problem as before, so we've got to remove the wiggly things. Where do they start? When I hover somewhere here, it is position 93, somewhere here. So let's remove things from 1 to 90. Delete 1 to 90. Okay, so now we're left with the prediction that seems to be of better quality. We can check with palette that's the case. Yes, that's things that are either blue or magenta with a little bit of yellow, but they seems to be uh, quite reproducible here. And if we color it by chain, here's another. Well, I'm gonna hide 3, 4 of those. I'm gonna hide this thing and color by chain. Now you'll see, so here are our 2 proteins, okay? Oh, I'm mutilated now, no. Back, I unintentionally cut out the other protein. It's not full version, so let's cut it out. I've got to close session file, open once more. I'm gonna read all 5 here quickly. Okay, now do tools and analysis and matchmaker and apply. Okay, that's good. Now it's better to color by chain. Okay, here's coloring by chain. I had 2 sequences here, so now I've got to be careful. Okay, so here we go. This is the starting part of the pink guy, so I've got to remove this thing. This is gonna be the signal peptide if present here. So this is 1 or so. What's the position here? This is 39, so let's remove 39, and this is chain B. You can see at the very end it's number 1/B, so this is chain B. 36, I'm gonna delete chain B from 1 to 36. Okay, here we go. Okay, so we trimmed the pink one, and now this was the thing that was unfolded. This is our signal peptide folding back. So now we're gonna get it here. This is 93. So here we can, and this is chain A. Okay, so that's a 1 to 90. Let's move to 990. Okay, so here we've got the misfolded, mispredicted parts removed. We can redo tools and look at structural analysis and matchmaker to align those. Apply. We've got a little bit better alignment in 3D, so we can color it by the AlphaFold prediction quality, and that's what we're getting here. And here's an example of pretty good prediction of a protein-protein interaction. We do have 2 chains. I'm gonna now hide all the copies other than the first one, so, and color it by chain. So here you see the prediction of a protein-protein interaction. We can show the side chains, okay, and color them by heteroatoms to simplify identification of the side chains. You do see for the purple chain the same disulfide as you've seen before. The pairs of cysteines are forming the right disulfide bonds to stabilize the pink structure. And then when you start looking through the interface between the contact sites between the chains, you might notice down here that there are a couple of phenylalanines that are buried inside the other protein chains. This is what you expect for hydrophobic residues to be buried within the structure. It can be either within a single chain, but they can be buried at the interface between two interacting chains. So this is quite sensible. You can notice that you have cases somewhere here. I can color somewhere here. Let's see that you've got positively charged residues here on one protein next to residues that are negatively charged on another. So here's a case of proteins side chains that interact electrostatically. This is also perfectly sensible and indicates that prediction is quite reasonable. And those are essentially things to look through when evaluating a prediction. You've got to look into different at each region of the protein and look if what you know about protein structure and rules and properties, that this is reflected in a prediction. If some of the things that you know should happen are not visible, or what you see violates your knowledge about protein structure, predictions probably flaky. The other thing to look at is how reproducible the prediction is. So if you see all five predictions generating similar structures, then it's another indicator that prediction is pretty good. And then the last thing to check is to look at another file generated by the AlphaFold. If we go here, there's gonna be yet another file. This is the one that it is here, okay, and that's what you see here. This is the contact map, the error in a distance estimation. You do see the first sequence. This is the short protein we've got there, and then the second one. Within each rectangle, those are the contacts within individual protein chain. So this is indication how good prediction is for the protein fold, for the structure of individual chains. So the large area with in blue indicates that those regions are predicted pretty reliably. The chain here is the one that I cut off the protein to, that this is the one that was unfolded. So this is what we do have in our final model. Same thing holds for the other protein. The other protein shows up here near diagonal, and this is the region that we left in a final structure. This was removed as poorly predicted with a single signal sequence somewhere there. The important thing for interactions is to look at distances between the chains. So that those are those two rectangles here and here. If you do see patterns of high quality predictions somewhere here, those blocks here, those blocks here, and those blocks here, they indicate that the relative orientation of those two chains is pretty well defined. This is indication that proteins were actually docked, and you can see that to various degree, the same pattern as you see here is repeated in every prediction, every version of the prediction. So overall, it means that the predictions is fine both for individual domains and the complex. The predictions where you didn't dock fine will have those regions here all shown in red as poorly predicted. So this is something that you should look at, and it gives you some information about the quality of the predictions. Okay, and that's about all we can squish in the class. I do have more predictions for other sequences that were predicted. So those of you who ask for predictions, I'll try to contact you and get you all the files that were generated. And if you're interested, we can meet on Zoom or in Boyer Hall to go over those predictions to see how to interpret those. And that's about it, I guess. That's the end of the class. If there are any questions, I'll happily take those now. So if you need some extra information, please ask. If not, we'll stop recording. Oh, here's a question on the chat, in the chat. Oh, there's a thank you note. Okay, so that's it. I'll contact those of you who provided sequences, and I'll drop all the files on the Google Drive together with presentation, so you'd be able to go through the results for the protein interactions. For the case like here, we've got where you've got two chains. Those things have to be generated on your own. So you'd have to either use public version of the server, or find someone me included who can actually run the predictions locally to see what can be done, because the protein interaction, the predictions of multiple chains are not available in a public databases. So they've got to be generated on your own. So that's how to deal with more complex cases. Okay. Otherwise, thank you for the patience. I'll get once more the QR code up, so that Eloy is happy. Where do we have it here? Let's see. Where do we have it? Gonna be all the way. Here. Okay, here's the QR code, and that's about it. So now I can stop recording. Okay.